Good morning and welcome to our webinar, Beyond Two-Target Staining, a practical guide to overcoming multiplex IHC hurdles with AtlasPlex. My name is Joel Svensson and I'm a product manager here at Atlas Antibodies. We are very delighted to have so many of you joining us today as we're going to explore some practical solutions within the field of multiplex immunohistochemistry. With us today, we have Mikael Malmqvist, our principal scientist. Hello, Mikael. And he's a key member of our development team behind AtlasPlex, and he will guide us through the technical challenges and innovations within multiplex IHC, sharing his expertise and insights throughout today's session. So we're very pleased to have you with us, Mikael. And before we begin, there are some practical notes I want to mention. Today's session is for about an hour, maybe less, depending on your questions in the end. And we're really happy for you to send us your questions, and you can use the Q&A feature in this Zoom webinar. And you can send your questions to us at any time, and I will gather them and we will have a look at them at the last slide. We really aim for an interactive session, so really look forward to your questions at the end. Also, to remind you that today's webinar is recorded and will be sent out to all participants after the webinar. So let's have a look at today's agenda. First, we will just talk briefly about who we are, Atlas Antibodies as a company and provider of the IHC portfolio, our legacy going back to the human protein atlas. And then I will leave over to Mikael, and he will take us through the background and reason for doing multiplex IHC and talk a lot about common challenges associated with different methods for conducting multiplex IHC experiments. And then we will have a look at the AtlasPlex technology, the kit itself, what are the main features and benefits with AtlasPlex, and also how to order an AtlasPlex kit. And then Mikael will finish off with showing us some real-world evidence of how AtlasPlex has already been used. And then we will continue and finish with a Q&A at the end. So again, thank you so much for taking your time with us today. We really enjoy this and I hope you have a good time. So before I leave over to Mikael, let's just shortly talk about who we are as a company. Atlas Antibodies, we were founded to commercialize the scientific achievements of the Human Protein Atlas, which is now turning into a 20-year mission to map the entire human proteome. And our IHC product line is built on this legacy, and we have been maintaining the highest standard for tissue validation, specificity, and reproducibility. So thanks to the Human Protein Atlas, we have this great antibody portfolio that was originally developed for tissue purposes. And that brings us to what we're going to talk about today, which is AtlasPlex, that really stands on this foundation of a great antibody portfolio that has been used for IHC, where we cover over 12,500 unique human genes, and we are now putting them into, packaging them into more advanced multiplex assays in order to generate deeper scientific insights. All of our antibodies included in the AtlasPlex have been developed by us and validated by us using immunohistochemistry, following the highest HPA standard for specificity and reproducibility. So that was a bit about our background. And with that, I will hand over to Mikael Malmqvist, our principal scientist. Hello, Mikael.
Hello, thank you, Ewan, for that introduction. Just bear with me while I swap the presentation over to my screen, just a few seconds. Hi, everyone. Thank you again, Ewan. A bit of background first. Classical histology and immunohistochemistry, IHC, to study how cells and their proteins are arranged and distributed in tissue are not new technologies. As early as 1801, Javier Bichat proposed that the human body is composed of distinct tissues. Albert Kuhns later on was a key figure in the development of immunohistochemistry. Lately, these classical histological techniques have been further developed to allow for multiplex spatial proteomics, enabling the simultaneous detection of several proteins in the same tissue section. Multiplex spatial proteomics has become one of the most transformative technologies of the decade, and last year it was even recognized as Method of the Year. So single and multiplex immunohistochemistry are two different approaches used in tissue analysis to detect specific antigens. Single immunohistochemistry typically involves the use of antibodies to detect one marker per slide, utilizing chromogenic detection methods. And this approach is straightforward and standardized, making it suitable for routine diagnostics. It has minimal impact on tissue preservation, but one drawback is that it only yields one-dimensional qualitative or semi-quantitative data at best, so it's therefore limited in analyzing complex tissue environments. In contrast, multiplex immunohistochemistry allows for the simultaneous detection of multiple markers on the same tissue section. And this is achieved through fluorescent detection systems, which require more complex workflows and careful optimization. So multiplex immunohistochemistry demands meticulous tissue handling to prevent degradation during these protocols as well. But when the workflow is optimized, it supports high-dimensional, spatially resolved data output. And this makes it particularly valuable in spatial biology, biomarker colocalization studies, and also studying complex cell interactions. So while routine pathology still largely relies on conventional single immunohistochemistry, multiplex immunohistochemistry is increasingly favored in advanced research and in clinical applications where detailed spatial and molecular information is essential. So why perform multiplex immunohistochemistry? Well, the human body contains roughly 37 trillion cells. So to translate data into patient benefit, we need to examine tissues and cells at multiple levels: tissue-specific, cell type specific, and subcellular, and then connect these molecular mechanisms to disease, biomarkers, diagnosis, and precision medicine. There are several approaches to perform multiplex immunohistochemistry with fluorescent detection in tissue. While these approaches enable the simultaneous detection of multiple proteins, many are associated with significant technical challenges, particularly when a manual workflow is used without the availability of a dedicated multiplex instrument. And I will go through some of these challenges now in the next few slides. The most commonly used method to perform multiplex in basic academic research is the indirect immunofluorescent approach, indirect IGIF. Indirect IGIF multiplexing is characterized by the need of using antibodies from different host species. The different primary antibodies bind the targets, and then they are detected with fluorophore-labeled species-dependent secondary antibodies. If you want to stain more than one target protein at a time, you need to find primary antibodies from different host species. Otherwise, you will get some cross-reactivity during detection. So, for example, you might purchase a mouse primary antibody from company 1 and a rabbit primary antibody from company 2, and then you may borrow a third goat primary from the lab next door. And you will also have to buy or borrow the secondary antibodies from alternative sources. Academic researchers often study diverse and novel targets, making it sometimes difficult to even find antibodies from multiple species. So when studying less common markers, researchers that do not have access to a dedicated multiplex approach, they will face challenges. There are, of course, species-independent solutions where you can use the same species primary antibodies for tissue multiplexing. One example is by labeling primary antibodies directly with fluorophores rather than labeling secondary antibodies. And this is known as direct immunofluorescence, direct IF. Alternatively, you can perform the indirect immune stain using primary antibodies followed by fluorophore-labeled species-dependent secondary antibodies. And then after imaging, you can strip the antibodies using heat to remove them before repeating the staining stripping cycle. While both of these methods help to avoid the species dependency hurdle, they do suffer from some major drawbacks, making them less suitable for protein detection in tissue. If you decide to use the direct IF approach, by labeling the primary antibodies directly with fluorophores, then you'll face problems with low signals because you hardly achieve any signal amplification at all. With the indirect approach that I mentioned in the previous slide, then you have several secondary antibodies that can bind to one primary antibody and therefore generate some degree of amplification. But with the direct approach, only a limited number of fluorophores can bind to one primary antibody. In these images here to the left, we have used our anti-6in antibody in testis. And one can clearly see how difficult it is to generate a decent signal to noise when using the direct approach compared to the indirect approach. The low signal to noise is particularly troublesome in tissues where you have relatively high levels of autofluorescence as well. Furthermore, the labeling of antibodies with fluorophores could affect antibody affinity, especially if the conjugation chemistry labels the antibody in a random, non-specific fashion. For example, many people use the method, it's called NHS ester chemistry, which covalently attaches fluorophores to amine groups on lysine residues of the antibody. And on average, each antibody contains 10 to 20 different lysines that are accessible for NHS ester labeling. And several of them are located in the antigen binding region, the Fab region. So essentially, the antibody can lose its ability to bind to the target protein. As mentioned, an alternative method to overcome the species dependency is to perform sequential staining involving repeated antibody staining and stripping cycles. In this method, one would stain the tissue with primary antibody followed by a fluorophore-labeled secondary antibody. And then after imaging, you will strip the antibodies before repeating the staining and stripping cycle. And one major drawback with this approach is that the antibody stripping can affect tissue integrity. The stripping is often performed using heat antigen retrieval treatment, which could damage the tissue, affecting the overall quality and integrity of the sample. And this is particularly noticeable when looking at smaller, delicate anatomical structures. This image here to the right highlights such a heat-induced tissue damage in the mouse skull. Higher magnification shows the delicate morphology of the endolymphatic sac, ES, with a monolaid epithelium enclosing the ES lumen. After heat-induced antigen retrieval to detect the antibody epitopes, one can clearly see an extensive disruption of the delicate anatomical structure, such as endolymphatic sac, indicated here by the arrow. Another drawback with this method is the relatively long time it takes to perform repeated staining and stripping cycles. A 5-plex experiment can take 3 days. So this is all from me right now. I will hand back over to you, Will. Thank you.
Thank you, Mikael, for that excellent overview. And let's now have a look at the commercial multiplex IHC solutions. And we're going to focus a little bit on the flexibility in target selection for panel design. So while researchers working in genomics and transcriptomics have access to solutions that allows for studying the full human proteomic length, as soon as we move into protein-driven multiplexing kits, those often limit the target diversity quite considerably. And this is probably because of the scientific-driven questions around the studying of tumor microenvironment that relies on a smaller set of common markers within cancer and immunology, but also because we know as an antibody original manufacturer, it's extremely expensive to develop and validating a broad range of antibodies. So as a result, most of the ready-to-use kit solutions on the market today, they are quite limited in terms of the targets that you can select for those kits. And they are also quite expensive, and mainly they focus, as I said, on specialized areas in cancer and immunology. And what we see as an antibody provider in the IHC field is a growing demand for a more flexible approach to designing multiplex IHC panels. And many researchers that use our products, historically, they have ended up manually building and optimizing their own panels and doing in-lab optimization. And this, of course, allows for a higher degree of customization, but it is rather time-consuming, and you will face some of those challenges that Mikael was talking about. And this is why we think there is a clear need for manual kit solutions, streamlined kit solutions that offer a greater flexibility in target selection. And this is precisely the gap that AtlasPlex was designed to fill in the market. So, and that leads us to the AtlasPlex kit solution. And I'm just here gonna briefly go through some of the key features and benefits with AtlasPlex kit. And we also gonna have a look inside the kit, what comes with the kit. And Mikael is then gonna help us understand more about the workflow and some real world evidence. As we have pointed out before, AtlasPlex was built on this rich foundation of IHC antibody portfolio that we got from the Human Protein Atlas. And this allows for an unparalleled flexibility in panel design, where one can freely customize panels from more than 12,500 unique human genes. But also importantly, we incorporate a TSA workflow into this AtlasPlex kit that better preserves the tissue integrity. And we're gonna look at that later. Mick is gonna explain how that works. It's a direct TSA workflow without elution cycles. And we also only incorporate trusted reagents into an AtlasPlex kit. So all of those antibodies have been developed for the Human Protein Atlas originally, and we maintain that high degree of IHC validation as a manufacturer. And we also use a workflow that comes with no species limits, and that is really what allows us to enable that wide range of products, considering that they were mostly derived from rabbits. And with AtlasPlex, you get great time optimization, both in terms of sourcing various reagents and optimizing them into a manual workflow. But also the workflow itself is something that could be applied in one day and get same day results. And as I said, Mick is going to touch on that briefly. So, and finally, as we're talking about multiplexing in tissues, it's very important with strong signal amplification. And we are using a pyramid signal amplification here that delivers strong, clear signal even for low abundance proteins. So when we say that we have unmatched or unparalleled flexibility in panel design for this streamlined kit, what is it that we mean? We say that we offer full customization of more than 12,500 unique human genes. And we just want to be a little bit more concrete about what that might mean. So on the left side, we just handpicked 38 of the most common immunology and cancer markers that are used all the time in multiplex immunohistochemistry and MIF. And you will recognize all these markers. And this is just to say that we, of course, cover these kind of markers with AtlasPlex. You might as well build an IO kind of panel based on immune cell lineage markers, functional checkpoint markers, famous tumor oncogenic markers, or structural markers. This is just a standard way of panel design. But the real beauty starts with what we show on the right side. And here we just cross-correlate the HPA data from single cell specific genes that are either enriched or enhanced in 81 different cell types in the human body, here visualized in 15 major cell types, just to show that one could find as many antibodies for highly specific genes that are enriched in those different cell types. So that gives users an incredible resource using the HPA also to design panels for an AtlasPlex kit. If you have questions regarding more scientifically driven questions regarding the how to build panels, etc., you can always call us, and we have great expertise in-house regarding both IHC and multiplex panel development, and we are more than happy to consult with you regarding those questions. So when we say AtlasPlex offers high flexibility, we don't really think that AtlasPlex is always going to be the perfect solution, but rather that the whole spatial field is a very wide and complex field, offering everything from the highly customized solutions on the left side here, where researchers are purchasing separate items and primaries and secondaries and reagent kits and try to optimize them in their labs. Then in the middle, we have some companies such as ourselves that offers ready-to-use kit solutions for either manual or optimized for automated workflows. We also have companies like Altiview and Cell Signaling that offers great ready-to-use kit solutions in this area with or without added compatibilities such as image analysis platforms. And then on the right side here, we have these fully automated systems. And maybe we can say that Akoya and other solutions are on that side. So of course, depending on the need for degree of plex or depending on the need for degree of flexibility, one should always tailor the multiplex strategy to match the need for scale, reproducibility, and flexibility. And this is where we think that AtlasPlex will have a place for some of these needs, especially when we talk about small scale, small to medium scale type of manual multiplex needs, but also AtlasPlex could be used in an automated way. So what really goes into an AtlasPlex kit? Let's have a look at that. So first of all, in our initial launch, we are focusing on maximizing the degree of flexibility in panel design and not necessarily maximizing the degree of plex, but we might extend this in the future. So we are now launching a 3 or 5-plex platform. And what you get here is the bioattenuated antibodies from us in 3 or 5-plex format. You will get the HRP labeling kit with horseradish peroxidase amplification and quenching buffers, as well as the tyramide dyes for either 3 or 5 colors. And we also offer different types of kits. So you can see either 3 or 5-plex, but those can be ordered as a standard kit, which means something that is enough for 25 slides, or one can choose the larger kit, which is enough for 50 slides. And this way, we try to offer some flexibility for users that want to just try something, initial studies, without committing to a more expensive kit for larger scale research. So yeah, so for kits, it's not so dramatic, but if you think about all the possible configurations from 12,500 choices, then it becomes rather challenging in what kind of panels one can order. So we had to come up with a way to help users configure the kits of interest. And I would just like to point out once again that you can always call us directly, and we're more than happy to help out with both panel design and setting up a kit order, whatever it might be. But this kit configurator tool, one can find on atlasantibodies.com, navigate your way to the AtlasPlex landing page, and there you will find this kit configurator. And it's a very intuitive tool that helps you configure and build a kit. And this can be used also for searching and exploring and comparing alternatives. So for instance, first you choose whether you want a standard or a large kit. You choose whether you want 3-plex or 5-plex. And then you will fill in as many different protein markers as the degree of plex that you chose. And there will be a preview button down here that allows you to look at the whole kit. And at this stage, there are no commitments or anything. This is just to have a preview. You can then see which antibodies goes into this kit that comes as bioattenuated antibodies from us. You will see what kind of HRP labeling kit and corresponding tyramide kit, depending on the choice. And you will also see a price down here. And as I said, this is something one can see if we have the ability of delivering such a kit, and also to compare price with other solutions. So feel free to contact us, or feel free to use a kit configurator on our webpage. I don't really have so much more to say about the product, Mikael. Maybe you can show us a little bit images, maybe, of the AtlasPlex, how it has been used.
Certainly, Joel. Thank you. Just bear with me again. I will transfer this.
No worries, no stress.
Right. So when we developed AtlasPlex, our goal was to overcome these technical hurdles with manual multiplex that I mentioned earlier, while still enabling the full flexibility in target choice from our extensive product catalog of the IHC-validated antibodies. So, as I mentioned earlier, one of the major challenges when performing immunostaining is that in tissues in particular, is to generate high enough amplification of the signal to be able to distinguish true signal from the background autofluorescence that is often encountered in tissue. So this makes it particularly difficult to detect the low abundant proteins in tissue. So to enable the large proteome coverage that Joel talked about, we needed AtlasPlex to utilize a detection method that allows for strong signal amplification to make sure that the product can be used to detect even these less abundantly expressed proteins. So therefore, we decided to include tyramide signal amplification with fluorescent detection, TSAIF, as the detection method for AtlasPlex. TSAIF generates a strong signal amplification by covalently depositing fluorophore-labeled tyramide substrate in and around the protein target. And this reaction is catalyzed by the horseradish peroxidase enzyme, HRP, which is then attached to the antibodies that you use for detection. So the standard TSAIF approach, see my computer's lagging a bit. The standard TSAIF protocol is to use an indirect approach where the antigen-specific primary antibody is detected by the secondary antibody, and the secondary antibody is labeled with the HRP enzyme. And this indirect approach extends protocol times because you have to do primary and then secondary antibody incubation. And then as well, with the standard approach, you can only stain one target at the time, because you need to perform these time-consuming repeated antibody staining and stripping cycles. And the reason for that is that you only use one type of label, the HRP enzyme. So if you applied several antibodies at the same time to stain more than one target, then there's no way to distinguish between the signals from different targets. So therefore you have to do the staining in cycles and then do this detrimental heat stripping cycle. AtlasPlex will overcome this problem by offering the next generation TSA-IF approach. And how does this work? Well, we label the primary antibodies directly with HRP. And this means that you don't have to perform the sequential primary and secondary antibody stain that is used with standard TSA-IF. So primary antibodies are supplied pre-biotinolated. And then they're labeled with HRP-bound streptavidin as part of the kit. So the user don't have to biotinolate the antibodies themselves. That is already taken care of by us. So the kit is supplied with pre-biotinolated antibodies. And then all you need to do is add the streptavidin HRP. And then HRP labeled antibodies are used for tissue incubation. And then followed by that, you do tyramid signal amplification. And this will result in a covalent deposition of the fluorophore labeled tyramid substrates on tyrosine residues in and around the target protein. And then the reaction is stopped by the inhibition of the HRP enzyme using a unique HRP quencher. And this is a key step, meaning that there is no need for any antibody stripping. There will be no negative impact on tissue morphology. And therefore you avoid this major challenge with loss of tissue morphology. And the use of directly labeled primary antibodies and the lack of this antibody stripping dramatically shortens the protocol times. So you can complete a 5-plex experiment in a day. So the workflow looks like this. First step involves labeling of biotinolated antibodies with the streptavidin HRP. Then you perform single target staining cycles involving antibody incubation, color development, and HRP enzyme quenching. And once the enzyme is blocked and quenched, the second staining cycle can be performed in the same manner, and then you can repeat these cycles up to 5 times for a 5-plex kit. Imaging is then performed after you've completed all the staining cycles on a standard fluorescent microscope equipped with filters that can separate either 3 or 5 fluorophores in the visible light spectrum. So as I mentioned, AtlasPlex utilizes the TSA-IF approach, which generates at least 10 times stronger fluorescent signals compared to indirect IF. Here we have used our anti-beta catenin antibody in rectum, and you can clearly see how AtlasPlex gives you much more signal to work with compared to direct IHC-IF or indirect IHC-IF. Beta-catenin is a relatively abundantly expressed protein, and this extra amplification generated with AtlasPlex may not be needed in this particular case. However, for other low abundant proteins, it could be more challenging. For example, when you try to detect low abundance neurodevelopment markers, that could be a significant challenge in both research and clinical diagnostics. Due to the low expression levels of neurodevelopment markers, and also different tissue specificity and variability across developmental stages. Here, AtlasPlex was utilized to detect double cortin as a marker for immature neurons in embryonic mouse cortex at embryonic day 11. And you can clearly see how the signal is amplified with AtlasPlex and how that improves the signal to noise in the double cortin stain compared to an indirect IHC-IF approach. You can also go 2-plex by staining an additional marker such as SOX2 to identify new epithelial cells. Or a few days later in embryonic development, on day 14, we can also detect early-born cortical neurons using our anti-TBR1 antibody in a 3-plex experiment together with anti-SOX2 and double cortin. Here, we have used the AtlasPlex to stain 5 targets in human cerebellum, analyzing the spatial distribution of different neuronal and glial cell types in the mature brain, including microglia, astrocytes, oligodendrocytes, and 2 markers for neurons. And if you wanted to map the cell type expression of your target of interest, you could include one or several of these markers in a AtlasPlex panel, and then you could just replace one of the markers with your target of interest. And mapping the cell type expression could be crucial for several reasons. Mapping the cell type expression for your target of interest. Different cell types in the brain have distinct roles. So therefore, knowing where your protein is expressed helps to determine the potential function of that protein. And also, many neurological and psychiatric disorders are linked to dysfunction in specific cell types. So for example, if your target of interest is expressed in microglia, that could suggest a role in neuroinflammation, which is often a hallmark of neurodegeneration. Here's another example where we have applied AtlasPlex to analyze FFPE sections for prostate cancer tissues. And we use markers for gland type, TP63 for normal prostate glands, and the MACR enzyme for tumor cells, prostate tumor cells. We also used immune T cell markers, CD3 and CD8. And first, we wanted to verify the expression pattern for our target of interest, which was keratin 5. Keratin 5 is normally expressed in basal cells of normal prostate glands. However, in more aggressive forms of prostate cancer, keratin 5 can be expressed also in tumor cells. Our observations here indicated that keratin 5 is co-expressed clearly with TP63, thereby confirming its localization within normal glands. Also, we could see that the tumor cells had no expression at all of these 2 markers, TP63 and keratin 5, showing that there was normally no expression in tumor tissue of these targets. And secondly, we looked at the tumor immune cell infiltration in the tumor gland, and indicated increased levels of T cells in these tumors. We also looked at the overlapping signal between CD3 and CD8, which indicates the presence of cytotoxic T cells if you have co-expression of those 2 targets. But we can also see that one of the cells here has only expression of CD3, so you could add an additional marker to investigate what type of T cell that is. For example, you can add FOXP3 to identify regulatory T cells that should be positive for both CD3 and FOXP3. FOXP3 was recently identified as a key transcription factor in regulatory T cells, work that led to the 2025 Nobel Prize in medicine. Right, so I will hand over again back to you, Will, for a wrap up, and then before we move on to questions.
Thank you, Mikael. So now that brings us to the end of this presentation. I should just change here, sorry. And before we head into Q&A, let's just quickly summarize some of the things that we've been talking about here today. On this slide, you will see a side-by-side comparison of the main multiplex immunohistochemistry methods as discussed here by Mikael. And what really stands out is that AtlasPlex with a direct TSA workflow consistently comes out on top on several of these key measurements, such as signal amplification is great, target flexibility in this type of product is unprecedented, and we introduce a species-independent protocol. It's very tissue preserving, easy to use, and the workflow is faster than traditional methods. But also, we priced AtlasPlex to help price-sensitive users in academic research to be able to afford these more complex solutions, and which is why you can always benchmark this against some other regular methods. So all in all, there are some compromises related to all kinds of different methods out there. AtlasPlex, together with this workflow, has been designed to help overcome some of these issues. And we think that can help researchers to go beyond low-plex, go beyond 2-plex stainings, allow for a third or a fourth or even a fifth marker into standard panels without adding too much complexity. So thank you all for staying with us during this presentation. And Mikael, now we have some questions. So let's see. Any advice for picking the right antibody combos from the Human Protein Atlas when building an AtlasPlex panel, especially for targets that aren't well studied? What would you say, Mikael?
One suggestion would be to use a panel that includes known targets, for example, cell type specific markers or tissue architectural markers. So, for example, you can use the markers for different cell types in the brain, or you can use tissue architectural markers like E-cadherin for that stains epithelial cells, collagen markers for like fibrosis. You can use CD31, which stains endothelial cells. So by having panels with known markers, and then you would then add an antibody towards your target of interest as a fifth antibody in your panel, for example, or as a third if you only do a 3-plex. So that would be my suggestion.
Okay, great. It's another question for you. Has the AtlasPlex kit already been evaluated for staining of circulating tumor cells?
No, it's not been tried yet.
Okay.
That's a good suggestion for how you could utilize AtlasPlex for sure.
Yeah. Here's another for you. What microscope setup works best for AtlasPlex? I'm talking filters, fluorophores, and if someone using an older scope, will it still work for 3-plex or 5-plex imaging?
Good question. I mean, for the 3-plex kit, we use pyramid fluorophores that like absorb light in the normal visible spectra. So 488, 555, and 647. So this means that any standard fluorescent microscope that are equipped with the 3 filter setup can be used. So you just need like filters that detect the emission in the green spectra, FFPE, or yellow spectra, which is Psi3 or TRITS, and then in the red spectra, which is Psi5. So and this 3-plex can easily be combined with the DAPI filter if you want to include a nuclear counterstain. For the 5-plex kit, that also includes the 430 and 594 fluorophores, and those two are fairly close to, 430 is close to DAPI, and 594 is close to, it sits between the 555 and 647, so that could have some bleed through into the regular filters. So for if you want to use the 5-plex kit, you need to make sure that you use narrow filters. For example, if you use a wide field epifluorescent microscope that do not have any bleed through across the filters, across the channels, or you can use computational software that many people use to do this spectral mixing of emissions across different channels. Additionally, there's one other option, is if you have a confocal microscope where you have tunable lasers or tunable detectors, you can, for example, a Leica SB8 or a Leica Stellaris, you can easily create your own narrow detector spans. So that would be for the 5-plex if you want to do that.
Okay, great. Have you noticed any differences running AtlasPlex on human versus rodent tissues, like in antigen retrieval or background staining? Any tissue-specific tips?
Well, no, we haven't seen any differences really. The key is that the antibody recognizes the rodent homologue, and if it does, then AtlasPlex should give you the specific information there as well. For all of our antibodies, we display the antigen sequence used when we generated the antibodies on our product, on the product page, and also we give the interspecies information on that product page. So you can see that many of the antigens that we've used have a high sequence identity to rodents equivalent proteins. So I think many of our antibodies will work in rodent tissue as well.
Okay, great. We have quite many questions, so really happy to see that, and we will try to do them all actually, but we'll see how much time we have. Here's another one for you, Mikael. How much primary antibody is provided with the kit, and what are your recommendations for validating and optimizing concentration of, in parentheses, these antibodies on our samples, for instance, single stains before multiplexing?
Yeah, so we provide enough antibody for each antibody in the kit. We provide enough volume to be able to perform 25 or 50 different reactions. And when I say one reaction, that's 100 microliter reaction volume. So that should be enough to cover tissue section, like a TMA on a tissue glass slide. That's the how much antibody you get. Sorry, I missed the other second question. Was it about the optimizing
I think
antibody?
the question was how much primary antibodies provided with the kit. And,
yeah, that was the first question.
Yeah.
Yeah.
And if there's anything you can say about optimizing concentration of these antibodies on their specific samples, for instance, single stains before multiplexing.
Yeah, definitely. You should always optimize, even though we give you recommended dilution that you should use the antibodies, that is always dependent on your tissue material, but also dependent on what instrument you're using, how much light do your different filters collect in the different wavelengths. So it might be that so first of all, you have to titrate the antibody and to optimize you get the highest signal to noise in your tissue, but then also you might have to, if you have in your panel one protein that's more lowly abundant, then you could put that in your filter where you collect the highest amount of light, so you have the highest sensitivity. So if you know that your FFPE filter is the best in terms of collecting all of the emitted light, then I would put my most troublesome targets in that channel.
Okay, great. Let's see. Can antigen retrieval be performed in the standard staining machines used in pathology labs, such as Ventana, Omnis, etc.? Do the kits come with suggested times for HIER and AB incubation?
Yeah, so for the AtlasPlex, we recommend to use the same antigen retrieval method that we use for our regular antibodies when they are used for regular IHC. So yeah, you can use any of those instruments that you mentioned there. Absolutely. Obviously, what we've validated antibodies in is our standard antigen retrieval protocol that we have on our website. So yeah.
Okay, thank you. Let's see, we have time for a couple of more questions, I think. We have really many questions, thank you. Some 10 years ago, we used tyramine biotin peroxidase from some vector ? With enormous gain of sensitivity over ABC and streptavidin biotin complex. But endogenous biotin was tremendous challenge, so we moved to polymer peroxidase. How can you avoid these problems? Did you get that, Mikael?
Yeah, so definitely. Yes, you do have some levels of endogenous biotin in tissue. And so what we make sure when you label the biotin-related antibodies with the streptavidin HRP, we make sure that any unbound streptavidin in the tube is blocked with a biotin block buffer. So we add excess biotin to the tube, which will block any free streptavidin. And that means that there is no streptavidin in your tube that can bind to this endogenous biotin in the tissue. So we haven't experienced any problems with background at all.
Okay, great. When you're trying to detect low abundance proteins in human tissue, are there any tweaks or panel design tricks with AtlasPlex that helps boost signal and cut background?
Yeah, I think I touched upon this before in one of the earlier questions. So any immune labeling approach requires optimization of your particular experimental setup. So it may be that your microscope filters enable higher sensitivity in a particular wavelength, for example, like I mentioned. So if so, then you need to then you can put the targets that are less abundant they should be put detected in the channel where with the fluorophore that matches that filter. In addition, the antibody dilution can be optimized to generate results with the highest possible signal to noise. We recommend a certain dilution, but it should definitely be antibody should be titrated to see what generates the best signal to noise in your particular setup. The concentration of the tyramid dye, we recommend a certain concentration, but you can if you experience high background, for example, then you can dilute the tyramid dye, so you use less of it, which will create less background, for example. So we have added a troubleshooting guide in the instructions for use with suggestions on how to optimize your protocol. So you can, when you receive the product, if you were to buy it, then you will have you will get the instructions for use together with that.
Okay, super. So a couple of more questions for you, Mikael. Can AtlasPlex be used with frozen section samples?
Yes, definitely. If you're, our antibodies are validated on FFPE tissue, so we don't know whether they will, whether all of them are working on FFPE tissue. Sorry, on fresh frozen. But because AtlasPlex is more lenient on tissue morphology, I would say that AtlasPlex, if you are to use fresh frozen, you're probably are better to use AtlasPlex than other methods that rely on this repeated stripping staining cycles. So I would argue that AtlasPlex would be more suitable on fresh frozen than other similar methods.
Okay, one final question for you, Mikael. Avidin has 4 binding sites for biotin, so primary antibody in the second round can bind to Avidin. How do you cope with this? Does the HRP quenching enzyme include both hydrogen peroxide and free biotin?
Well, without going into too much details on how the Avidin that we are using, the chemistry behind it, and so on, there won't be any free Avidin sites available after we've added it to the antibody. We're using monovalent streptavidin.
Okay, I got some information here that I actually missed one question here. Sorry for that. We have, for labs new to Multiplex IHC, what kind of hands-on support or troubleshooting help do you guys offer when setting up AtlasPlex panels? I think we touched on this before, but maybe you can give some.
Well, yeah, like you mentioned, we will definitely offer some support in panel design. If you contact us and have a specific research question, we can help you to design a panel that would help you answer that question. But then, of course, we give you technical support to help you optimize your particular protocol as well. If you have any technical questions about setting up the experiment.
Yeah, super. Yeah, I got some instructions here. I can see only one more question, so I will take that one. Regarding kit size, what does 25 RXN mean really? Not sure about RXN. How large is the area of the slides?
Okay. When I answered
reactions, sorry.
Yeah, that question before is that one reaction is 100 microliter reaction volume.
Yep.
And that covers, what's our TMA? Is it, I need to talk to people in the pathology lab, but it's, isn't it an area of 1 × 1 centimeter or something? Or 2 × 2 centimeters tissue area. So we put a pap pen around it, the tissue TMA, and it covers that easily. So definitely, so 100 microliter will cover your tissue TMA that consists of 24 tissues or so, something, tissue cores.
Right. Hope that was an answer for you. So yeah, I cannot see any more open questions. So we would like to thank you so much for taking your time with us here today, and hope that you learned something new regarding our AtlasPlex new offering, and hope that you liked it. And as I said before, please contact us directly if you have any more questions. And I should also show you this. You can contact me or Mikael directly if you have any specific questions around the AtlasPlex, or you can reach out to our support at atlasantibodies.com, and we are always there to help you in any way we can. So with that, I would like to say thank you from me and Mikael today, and hope that you have a nice day. And yeah, thank you, and goodbye.
Thank you.
Thank you, Mikael.
Thank you.